All very quiet.
I did put them all on mute. That was me.
Well, that's good. Mm-hmm.
Hi, everyone. Thank you for logging on. We'll just sit about for a couple of minutes and give people some time to log in, and then we'll get started. Alrighty.
Some background music playing.
Yeah, we might kick off. We've got a good amount here to begin with. So, thank you everyone for coming on tonight to our webinar, talking about foliar testing with Graham Lancaster from EAL. So Graham is, as I said, from EAL, Environmental Analysis Lab. Graham was one of the founders of the lab, and they offer a range of testing. He just said there's upwards of 3,000 tests you can do through the lab, including soil and tissue and sap, and as well as quite a few others. So I'm Hannah. I'm part of the Sustainable Ag team for Hunter Local Land Services. We've also got my colleagues, Sam, who's from the Lower Hunter, and Maria from the Upper Hunter on tonight.
For everyone online, I assume you can hear something, so mic should be working.
Yep.
Thanks.
No worries.
I could just imagine a few people there panicking, going, I can't hear anything.
No, all good. So this webinar is actually going to be the forerunner for our upcoming foliar nutrition workshops, which are going to be held across all 3 areas of the Hunter region. And they're going to be run with Joel Williams from Integrated Soils and Grant Sims from Down Under Covers. We do have a date change for these events. Unfortunately, Joel had been emailing us with February dates, but was actually meaning for them to be in March. So you will see an email come through to you guys ASAP for those date changes. And hopefully you can still come along. So I'll just start with an acknowledgement of country. So we acknowledge the land that we come and meet on tonight. There'll be a few from different areas. So those are the new dates for the Foliar Nutrition days. They'll come up again at the end of the session, so you'll be able to see them while we're all logging off. So we're now in March, starting with the Lower Hunter on the 16th, Upper Hunter on the 17th, and Manning on the 18th. So for some housekeeping today, so this is all recorded. It's currently recording now. It's going to be uploaded on the Local Land Services YouTube channel so that people can come and watch it at a later date. We have set you up to use the Q&A function to ask any questions as the presentation goes along. So please use that. It's at the top of your screen. So type any questions you have in there. When Graham's finished presenting, my wonderful colleagues, Maria and Sam, will read out any of your questions that you've asked along the way to Graham, and he can answer them. And if we have time and get through all of those, I'll let you unmute yourselves and we can have a bit of a discussion as well. Currently in the Q&A is a link, which I now realise just looks like a link to nowhere. It's actually to a feedback form. Yes, I can hear the groans through the silence. I know they suck, but they are really helpful for us to get a feel of who you guys are online tonight. If we've hit the mark, if there's something that has helped you, and what else you might like coming out of the pipeline from Hunter LLS. So if you could just take, the 2 to 3 minutes to complete it, you can do it as we're wrapping up. It will also be sent out via email. So anytime you can do that would be great. Alrighty. So I'm going to stop sharing and we're going to hand it over to Graham, and he's going to get started with his presentation. Thank you. All right, Graham, you're right to start sharing.
All right, I think that should be showing now.
You're up, perfect.
All right, thank you, Hannah. Thank you everyone for joining tonight. As you know, this will be recorded, and I'm also happy for Hannah to share a PDF of this presentation. I do have some data in there and some information and some guidelines and a few other things that hopefully might be of some use. So as we go through it, and like Hannah said, we'll have plenty of time at the end for questions. So I'm the Senior Manager at the Southern Cross University Analytical Research Centre at Southern Cross University, or effectively Environmental Analysis Laboratory. My contact email's there. If you do send me emails, just send it maybe once or twice. I don't get to all my emails, but that's just part of it. So a little bit on EAL. EAL is Environmental Analysis Laboratory. We're an independent university-owned research, teaching, commercial analytical laboratory located at Lismore, Northern New South Wales, but servicing all Australia and international. We are getting more and more international samples these days. Founded by myself around 1992 after studying environmental science at the university here. And seeing all the problems that agriculture effectively was subject to, and pollution and over fertiliser and over pesticide and some issues there. So started up the lab. Self-funded facility. We now have got over 70 staff and turning over about $10 million a year, plus a lot of research work. Quality assurance, we have NATA, National Association of Testing Authorities, and ASPAC, Australasian Soil Plant Analysis Council, and I'm also the New South Wales rep for that organisation. We have a large range of services, so we're not just a leaf lab. We do a lot of soil, a lot of water, compost, biochar, hair for human nutrition, liquid and solid fertilisers, so quite a lot there. State-of-the-art equipment and instrumentation. Basically, this is what runs our leaf analysis. We have these LECO TrueMac CN 928s and CNS 928s. We have about 6 LECO combustion analyzers, and they typically run your carbon, your nitrogen, and your sulfur. For your nutrient analysis, we have these FIA labs down the bottom right-hand corner, and these generally run phosphate, ammonia, nitrate, and nitrite on leaf extracts, which is the sap, waters, and soil extracts. We work there. The only way we do leaf analysis, we routinely use inductively ICP OES, inductively coupled plasma optical emission spectrometry for soil analysis. All our leaf analysis, we go straight to the mass spectrometry, which is a superior piece of instrumentation, about 300,000. We have 3 of those that routinely do leaf analysis, whether it's SAP analysis or the digests. Those actual digests you can see on the bottom right there, they're acid digests of leaf tissue samples that are going through the mass spectrometer at the moment. And that's the sort of extracts that we run. They're quite a clear extract. So what to look for? It's about getting the correct leaves when you're collecting your leaf analysis, and I'll talk a bit on that, because there's mobile elements and they vary between which leaves you get on the tree or whatever crop you're working with. You do have to be aware of that. Field contamination, soil, dust, fertilizers, pesticides. If you've just sprayed, they often have some sort of nutrient base to them. So you've got to be careful of all sorts of spraying. You can ask the lab to wash the leaves or not. It's an extra few dollars, and it's an option there. And it's just up to you whether you think you need that done. Fresh leaves, we want to see them fairly fresh. It's the best analysis if they haven't sat in the post for 2 weeks. So generally try to post them often on a Monday or courier them on a Monday or a Tuesday, so they're not effectively sitting around, particularly in summer on the weekend. Sorry. Grinder contamination is something labs have got to watch out for. And when you get your results from labs, just be aware that there's always potential for contamination, even though most labs work really hard these days. And the weight you're getting, the results you're getting are dry weight. We dry them effectively at 7 degrees Celsius, and that's how the analysis is conducted. It's on a dry weight, so it's standardized to a dry weight. So when you're talking parts per million or milligrams per kilogram or even percent, it's always on a dry weight basis. So if the leaves are wet or dry or whatever, it's always on a dry weight basis, and I'll talk a little bit more on that. So this is just a bit of a publication. It's 95, but it's still fairly relevant. Sampling can be up to 1,000% error, which is quite extreme. If you don't get the right samples, it's some real issue there. Sample prep, the labs have got to do it properly, and they've got to put a lot of effort into how they prepare the samples, how they dry them, how they grind them. We grind them to a fine powder in special stainless steel mills to ensure we don't get any contamination or any cross-contamination or any mix-ups. There's a lot of money and effort goes into that. Instrumental measurement, like I said with the ICPs, normally only 10. It says 20% error, but these days it's 2 to 5%, maybe 10% potential error there just on the instrumentation. And data evaluation, everyone looks at the data differently and interprets it differently, so there's a percentage error there as well. So it's not the perfect science, but I'll explain where it's relevant and where it's not, to hopefully give you more insight into it. When I said mobile elements, nitrogen, phosphorus, potassium, magnesium move readily throughout the plant. The variable mobile are sulfur, copper, zinc, and molybdenum, and the immobile are the calcium, manganese, boron, and iron. So what that means, if you're collecting, say, the new growth leaf and the last year's leaf and the older leaves, the calcium, manganese, copper, boron, iron will probably be similar between each of those plant materials. However, they get more variable. Often in the new growth, the nitrogen can be 5 to 6+%, depending obviously what tree, what plant, and all that sort of thing. It can be a much higher in that new growth than the older growth. and same with those other phosphorus, potassium, and magnesium. So you've got to be aware that these things do move around. So when a guideline or a specific part of a plant is required or suggested for collecting, you're best to keep to that guideline. And that impact will be mostly shown in those mobile elements. I hope that makes sense. We have a bit of an information sheet on our website, just really the basics on collecting and sending leaf samples. The SAP is a little bit different. We don't have a separate information sheet on that, but you don't have to collect those in a paper bag. You just need enough SAP, typically in a plastic bag, because we want to maintain the moisture, that we can press them, and we use a hydraulic jack to press them and get the sap out of those. So we have quite an elaborate setup to press them. Often what we do, and mostly what we do, as soon as we receive your sap, we actually put them in the freezer overnight, and that breaks up the cells in a lot of those plants, because a lot of plants don't actually produce that much sap. And then the next day we thaw them very quickly and put them in a press, and then we get often just a few drops of sap out. And that's how the sap analysis is done. The leaf tissue analysis is quite different. They are often sent in paper bags because we don't want those too moist. Like I said, we'd run them on a dry weight basis. And we run them so that we immediately wash them when they arrive. And we put, if they want washing or requested washing, we then put them in a 70-degree oven overnight. In the morning, they're crispy, dry and very breakable, and that's when we put them through the grinders. Sometimes we have clients, particularly overseas, that we get them to dry it in any oven, 65 degrees, a home oven or whatever, just to preserve those samples. And once they're dried, they're actually preserved for weeks or months, because that drying process takes away the moisture, takes away the microbial activity, and really preserves those samples. And obviously with SAP analysis, you could freeze your samples there to preserve them and send them frozen, or send them as they thaw. And that could be another option there. And just note to the lab that you're doing that. Typically in our information sheet here, it says minimum 100 grams required. The reason we say that is we want to get a representative sample. So we want lots of leaves, and I'll mention this a bit later as well. But we actually only need very small quantities. For the instrumentation that we've got, we can get away with a couple of grams of sample, effectively a couple of leaves. I should be cautious on saying that. We don't want you to send a couple of leaves. It's really beneficial for research, because in research, they often have seedlings and they want to know what's going on. And now researchers will send us hundreds of samples of just a couple of leaves as seedlings grow and that sort of thing. So it opens the door on research. It can be used in the commercial world as well, and for when you're doing your own research. But it does show to you that we can get away with small amounts, but we do need a good representative sample. When we get 100 grams, we dry most of that. We crush most of that, and then we homogenize all of that to get a good representative sample that we then subsample from for the analysis. With sap, I've mentioned before, we only get 1 or 2 mils at most of sap. Some trees like macadamia in our region up here in northern New South Wales, the macadamia leaf just doesn't give much sap at all. It's a real pain for us to analyse. So for sap analysis, it's really only particular crops are good for it. And you've got to see what data is out there for interpreting the sap. But the leaf analysis is still the most standard. We do maybe 5 times as much dry leaf analysis than we would sap analysis. But maybe 2 years ago it was only 1/10 of the samples were sap, or 1/20 of the sample. So sap analysis is getting more and more popular. But I don't think there's still a lot of data on interpreting sap, and I will go into that in more detail shortly. This is the dry guidelines. So for dry leaf analysis, CSIRO put all the work into this plant analysis and interpretive manual. Ruder and Robson. This is the second edition in 1997, and this has excellent data for interpreting. And this is what a lot of the agronomists use, and a lot of the fertility reports you get done use these guidelines. And I'll briefly go through some of this. It's not a cheap book, so it's not something you're going to rush out and buy. But we're happy to scan in pages and send to people if they want information. You can see here as an example, this is alfalfa or lucerne. It has just pages and pages of all different crops, from flowering crops right through to native species, through to vegetables, etc., tree crops, forestry, etc. Here we have pre-flowering so I'm saying which part of the plant are you taking. A lot of the guidelines have a different part of the plant with a different guideline. So pre-flowering, first flowering, early flowering, hay, vegetative first, branching, budding and flowering. That's the most comprehensive sort of list of plant parts for the lucerne there. Most of them maybe don't have that. They might just have a particular part of the plant they want you to collect. I often say this about what you're collecting is what you're analysing. So it says collect a certain part. If you're doing your own research, like comparing different paddocks, then it doesn't matter what part you collect as long as you collect it consistently over both paddocks. So if you've got a good performing paddock and a bad performing paddock, you could collect the fresh growth on one paddock and fresh growth on the other paddock and do a direct comparison of those results. That's fine. That's your own data. You may not have many international or national guidelines to compare it to, but when you're doing your own research on your own farm, it's about building up your own data. It's really the sky's the limit on where you want to go with it, with the analysis. So in this summary CSIRO guide, it has general description of symptoms of nutrients deficiencies. They're quite a extensive guide, but it doesn't photocopy well. So what I did is got AI to summarize these tables. So this is just a summary of the common nutrient symptoms: nitrogen, whole plant chlorosis, yellowing, older leaves affected first, and specific examples there. So there's quite a lot of detail here that AI is extracted from there, and I think it's a really good summary. And that's why I said before that I'll provide this to Hannah so that you can share it, so you can have a read through it and just look at this, and hopefully it'll give you some benefits there. It's not the original, so I'll put that disclaimer in there. It is a summary from that reference I showed earlier. And if you want deep pages of that reference, I can share those as well, or you can buy the book or whatever. But that's there. But then also further, there's common toxicity symptoms. And these are related to ammonia, phosphorus, salt, sodium and chloride, manganese, zinc. And then there's some additional ones there with nitrate toxicity, sodium sulfate, chlorine or chloride. They're often used interchangeable, those words. Aluminium, boron, nickel, and fluorine or fluoride. Once again, that's used interchangeable. If you see the word fluorine, it's often talking about fluoride, and chlorine is often talking about chloride. It's just some chemical terms there. So that's more of a reference guide. Plant leaf sampling. Now, plant sampling is often yearly in autumn and spring. So often people that really get a lot of benefit out of leaf testing, they might just do it yearly, and they'll do it in a certain month and consistently do it over a number of years to build up their data set. Or it might be spring, but often they avoid the real cold winters and the real hot summers, depending on where you are in Australia, obviously. Generally early morning, particularly for sap analysis, is important. You don't want the leaves all dried out. So before 8 a.m. or earlier, most people do that sap. And a lot of the regenerative farmers will have their own meter there to work out the sap, some of the bricks testing, and maybe a little pH electrode to do their own monitoring there, and then send a sample through or whatever. For plant parts, I got to emphasize there, refer to the CSIRO Plant Analysis Guide. It has chapters on plant parts, but generally it says recently matured leaves are often considered sampling the third whorl back on branches, which is tree crops. And that third well back is not your new shoots and not your old shoots. With pastures, it's your whole shoot is often sampled. And with the grapes and citrus, it's your petioles, your leaves there. Most important to collect a representative sample, multiple trees, multiple plants, many samples from a pasture. It might be a half dozen or a dozen samples from a paddock, mixing in a bucket and then taking a handful of that out with gloves maybe, and putting that in a paper bag. Decide if leaf washing is required in the lab. So that's your call. We don't want to wash every leaf because it runs a risk that we could contaminate it, even though we're very, very careful there. So it's only really recommended where you think it's needed, and it obviously costs you a few extra dollars as well. Decide if some pre-drying is beneficial for you. So if you want to pre-dry them just so you can collect them from a number of paddocks and send them once every 2 weeks or once every week or whatever. And if your postage is slow, then pre-drying may be an option just to preserve the samples that I've mentioned before. Now, benefits of monitoring leaf versus soil. It's one of the really big questions. Why test leaf? This is my version. It's an indication of plant nutrient uptake. So you can have the most perfect soil in the world, but your plants may not be taking up those nutrients. And there could be many reasons for that: biological activity of your soil, suppression by high clay or high pH or low pH or whatever. So it really indicates your nutrient cycling, what's getting into the plants from that soil. It's ideal for assessing nutrient toxicity or deficiencies. So soil, you may have the best soil in the world and balanced and all the rest, and everything's not performing as it should. So that's where you may want to look at leaf net testing. And I'd start with leaf tissue testing and then look at sap later, just to see if you've got any nutrient toxicities or deficiencies there, to make sure the whole system is functioning. And it's great for paddock and crop comparisons. Like I mentioned before, if you've got a bad paddock and a good paddock and a not so average and all range, then you can really collect the same samples from each of those and directly compare them and see what is cycling into those plants. Why test nutrients? Well, testing nutrients, really trying to balance that soil, looking at what nutrients are lacking in the soil. If you haven't got those nutrients in the soil, then it's very hard for the plants to have that cycling and get it into the plants. So you've got to have a good balanced soil. You want to determine what excess nutrients are in the soil, because that could push excess nutrients into the plant, which then causes deficiencies of something else. So it is quite a balanced system. And that's why I talk about nutrient balance. If you have excess phosphorus, and I do see this a lot in the soil, then the potential there for the plants to take up too much phosphorus and have a deficiency on something else because they've got a preferential forced feed effectively happening. Relate chemistry to physical characteristics. So a lot of the chemistry in the soil can affect the compaction of the soil. The cation balance can affect the soil characteristics. And directly target fertilizer or compost application. So you're not just putting on broad range of fertilizers, you're putting on just what you need and not what you don't need. So that's the benefits of soil testing, and hopefully you'll understand a little bit more on why you might do some leaf testing. What's the difference between leaf and soil analysis? Typically in soils, I often have this diagram where I look at the soluble nutrients, which is typically your nitrate, your phosphate, your ammonia, your sulfate, those sort of elements, even potentially your chloride. You have your exchangeable elements, which is your exchangeable sodium, potassium, calcium, maybe a Bray1 or a Bray2 phosphorus. And then you have, we often test in EAL, the total nutrients to see what store of nutrients are in your soil. In a leaf tissue analysis, it is total. It's this total digest we do on the soil is the total digest you do on your leaf testing. So most of your leaf testing is totals, and your results are as a percent or as a milligrams per kilogram dry weight as a total. Some elements, optional extras effectively, are nitrate, ammonia, so some soluble phosphate and chloride, which are your soluble elements. So it's sitting more in this pool here of soluble elements in the leaf test. But like I say, normally it's a boiling acid extract that we do on the leaves that's dried and ground, that we look at the total content. Sap analysis is this orange box here of the leaf test. So it's just taking the sap and looking at all those really soluble moving elements in that sap, and that's all you're looking at in the sap analysis. So it's only part of the leaf, and it's only the part that's moving around the plant, the sap. So quite a simple explanation, but that's all it is. That's the differences. And like I said, recommend initially definitely going with your leaf tissue testing if you're moving into leaf testing. X-ray fluorescence. XRF is what we often talk about when we're trying in science, in doing studies, look at what the total constitution of a soil is. And when we look at that, your soil is mainly constituted or contains oxygen, 44 to 49%, silicon, 22 to 36% as silicon oxide, aluminium, 6 to 10%, noting 1% is 10,000 milligrams per kilogram. And then you go down and your carbon may be 0.1 to 5%. So you get a mass balance of 100% effectively in the end, but your main nutrients are really only a couple of percent. It's the same in a plant. This is an X-ray fluorescence of a plant, a number of plants there just as an example, where that's in milligrams per kilogram. This is a tissue analysis by portable X-ray. And you'll see here, if you add this up, that's potassium's 1.4%. You're only talking a few percentage of that leaf constitution is actually nutrients. A leaf is about 50 to 55% carbon. It might be 20 or 30% silica. It has a bunch of other elements, mainly the silicon oxide. So your oxides will be in there as well, just like the soil. And then what's left over is these are the nutrients that we're talking about in analysis. So when you're trying to look at mass balance, there's a lot missing there that you're not testing. We don't need to test the silica. Often we will provide the carbon of a leaf test as it's part of the LECO combustion results. So we'll provide that, and some clients use that. And to do a proper leaf silica test, the acid digest will not break up that silica. So you may get a leaf acid silica result, but to get a proper silica result from a plant, we've got to do an X-ray fluorescence, or we've got to do a fusion at high temperature with an alkaline material. So sorry to get complex there, but don't rely on the silica typically in leaf tests. It's not a reliable number. So this is the end result you get when you get a plant leaf testing option from EAL. These are our packs, our plant packs. That's our retail price XGST. It's a PA pack 001. Will give you total nitrogen, total carbon, total sulfur, sodium, potassium, calcium, magnesium, phosphorus, silicon. And like I said, it's not a true silica, it's acid extractable silicon, cobalt, molybdenum, zinc, manganese, iron, copper, and boron. And that's what you get as totals in that whole leaf test. Optional extras is in this other suite, which is a nitrate, a chloride, and yeah, they're just an extra, and ammonia that runs in there as another suite, or you could just add on a chloride or something like that. But that's a PA pack 008. Leaf washing, an SSPP 009. We have these codes that sit in our lab information management system to enable us to book them in quickly. That's $12 retail rate there, XGST. So you can see it's quite a lot of work for us. We've got to wash them in a detergent, and it does take us quite a lot of time to wash a single leaf sample. There's some results there. This all comes out as a PDF and a CSV file, which can be opened as Excel. So you can then use the data or manipulate the data or use plot the data or whatever you like there. We have our units, our element down here, our units, and our method reference right through there. And that just has some details there. We have some ratios, so nitrogen to sulfur ratio, nitrogen to phosphorus, nitrogen, potassium, which are good indicators. And obviously up here, carbon, nitrogen, crude protein, which is a calculation based on your nitrogen. So that's the standard report format that you'll always see coming out every time on a leaf test. On a SAP result, that's our SAP. It's a little bit more expensive. It's a PA pack PK 006. It has slightly different elements, and like I say, they're soluble elements. Has a moisture content, which I've always found important to provide the moisture content of that leaf, the leaf sample we're analysing. pH, a conductivity, which is a salt content, total nitrogen, a nitrate ammonia chloride. your calcium, magnesium, sodium, potassium, phosphorus, sulfur, boron, copper, iron, manganese, zinc, aluminium, molybdenum, cobalt, silicon, and bricks. We provide a bricks reading there that we put a little bricks meter there. Once again, you can get a leaf washing on a sap if that's an issue, but it's 50-50 how many clients sort of get that done. Once again, a standard format, with your results up here, your element, and this time you're in units, milligrams per litre of sap. A litre of sap would be a lot, but that's the standard units. A milligram per litre is a parts per million, or ppm. It's the same unit. If you see that ppm or milligrams per litre, it's interchangeable. So effectively, we take that 1 mil of sap and then work the results out in milligrams per litre. And you can see here nitrogen, it's your high one. Sodium, potassium, etc. I won't go through all those elements. Through to nitrate, ammonia, chloride, your pH, which you should know, 6.1, 3.98. Two plants there quite different from young and old. Bricks reading 2.8 and 5, and that can go right up to about 18, I think, of bricks, or 15. Your moisture content, so a leaf in this particular case for a potato is 89.3% moisture. So it's only 10.1% dry matter. So it's quite a moist plant. And we just let you know whether you requested or we did leaf washing. And that's a yes there, saying the client requested or we did it there. Then we have an option where we work with some agronomists to provide an agronomy report. It's another optional extra. We use Fertitech as one of our suppliers for agronomy reports. And we just send that off to them and they come back with a report to you. It's a PASG Sing 002, and we say there what it is and $55, and that comes back in a few days after we send the results through to them. And they then look at the cucumber, the guidelines from CSIRO publication or some other data that they've built up, and they look at whether it's high, low, or indifferent, whether it's deficient, adequate, high, or excessive. So you can see there quite a simple explanation. Now, you may not want this on all your samples, but you may want it on just a couple of samples, so that it's always an option. If you're doing quite a number of paddocks, one of the paddocks, you might just get a fertility report, and that'll give you the guidelines and what's high, low. So you can see here the desirable there, 5.5%, and 3.76% found for percent nitrogen. You can see through there. We also have that, on that same report, he often has comments like nutrient cycling constraints, nutrient ratio and balance issues, but often he won't actually give you folio options for how much foliar spray to add. This particular agronomist is more, he emphasises that soil is where you should be adding your nutrients. And here's you're just looking at where your deficiency is and then looking at your soil to work out what you need to add in crop-wise. There are agronomists out there that will give you foliar options, many agronomists, but this one here particularly doesn't do it at the moment, so I just want to give that disclaimer there. On sap testing, same thing. He has guidelines on expected sap for cucumber, and those are provided in the report there. It's a different code, PASG 003 instead of 002, and it gives you a SAP report, and that gives deficiencies, acceptabilities, or excessive levels. And obviously you can see there, this particular client has some serious chloride issues, 4,400 ppm or milligrams per litre. And you'd probably pick that up quite readily in the soil and the tissue analysis. I haven't really mentioned, and it's a very hard one because you'll probably ask the question, is there a direct relationship between soil, leaf, and sap analysis? And I don't think anyone's really done the work to find that. I think that's up to individual clients and agronomists to try to work that out. But I wouldn't say there's a direct relationship between any of them. I think if one indicates high phosphorus in the soil, it's most likely you'll find that in the sap and in the leaf as well. But there's no guarantees. Like I said, it's all about cycling of nutrients and whether they're bound up or whether there's preferential transfer of nutrients through the plants, that sort of thing, or preferential uptake of nutrients. We're not dealing with simple systems here. If it was simple, then everyone would be doing it and farming would be easy. So really, that's all I've got on this little presentation I whipped together. and hopefully it gives you some insight into plant foliar analysis and also sap analysis. So we can go with some questions.
Very good. Thank you, Graham.
Would you like me to stop sharing?
No, you're right. Oh, yeah, actually, you might as well. I don't think we've had any questions come through the chat. I'll just let you all unmute yourself if you want, and just put your hand up if you've got a question. Dale, I can see you do.
Hiya. I actually posted 2 questions or 3. Don't know why they didn't come. But anyway, I'm happy to talk out loud. So you partly answered the first one. Thank you. What determines whether you sample leaf or sap? So that's fine. And I put a rider in there that we send leaves off every year for PDL analysis, taken opposite inflorescences. So I assume that works for grapes. That seems to be the and I do the same every year, so I can get some consistency. Also, do you have a comment on the usefulness of applying crushed crystalline rock like basalt? I know one of the universities is doing a trial on it at the moment. Is there anything happening there?
Yes, look, I do think it has a place, but definitely not in elevated. I often find in farming that people think that a little bit's good, so a lot must be great. So I think very finely found, because basalt is a very resistant rock. It needs to be very finely ground and very lightly distributed. And I often find if you can put it in compost or organic matter and sort of give it some basis, I think that works very well. And it probably the same applies to biochar. In small doses, I think it can do wonders. We look at the reactivity of the rock. I can't even think of the unit at the moment, but we've looked at that, and it does have a lot of magnetic properties as well. And it obviously does have very good phosphorus and other elements there that just seem to work. So I think it is beneficial, but I'd always emphasize to people, just try it yourself and just don't spend much money on it and just give it a go in part of your paddock. I think farmers need to be scientists as well and do their own science and not just rely on everyone else, and just try it on part of your property without damaging anything or going high risk. Just give it a go and see if you get the benefits.
Yep, I'm keen to find out because it's cheap.
Yep.
The other question I had is, would you like to comment on whether fertigation or foliar application are more effective?
Look, fertigation. I assume with fertigation you're looking at the soil around the base of the plant.
Yeah, through drippers. Yep.
Yeah. Look, I like to feed the soil, but I think if you do start looking at deficiencies or toxicities or issues, then the foliar is a definite way to fix up things quickly. Like I said, I like to feed the soil and get a really balanced soil with a good carbon content and good biological activity, and then go from there and use the foliar if you've really got to sort out some issues on the health of the plant or whatever. So I'd be generally along the lines of feeding the soil.
Right. Thanks, Graham.
Brilliant. Thanks, Dale. Yoey, you've got your hand up. Go for it.
Yeah, good. Thanks very much, Graham. Just a quick one for us farmers out in the paddock. Have you got something other than bricks we can be using to give some sort of more immediate analysis or assessment about what we can be applying? Is there some simple field tests that you recommend?
No, I wish it was that simple.
Yep.
I've been to various conferences over in California and South America and also Netherlands on agronomy and soils, and they're starting to come out with these NIR units, near infrared, but don't get suckered into them. They have a probe that you can walk around push into your soil and it'll give you your phosphate, your nitrate and ammonia. And without swearing, it doesn't work. I haven't seen any of them work. And they have beautiful brochures that even I look at and go, wow, that's amazing technology. You've got to buy one of them. Don't get fooled by it. So there aren't any simple tests. The only simple pros we know of are pH, conductivity, your bricks, your temperature, your carbon dioxide for compost heap. There's certain things that we know are reliable. But when it comes to looking at your phosphate, your nitrate, or your total nitrogen, or your biological activity, I think you're really pushing uphill there. However, in soil care, which I'm a big part of soil care in this region, and soil care is mainly North Coast here, they have soil health cards. And I often recommend people to look at soil health card because it starts looking at how you can look at your soils yourself by a 10-point system where you're pushing a probe into the soil, you're digging and looking for earthworms and all this stuff just to look at your soils yourself. It'll show more structure than chemistry. It might have a bit of a pH test in there and that sort of thing, but there's no simple solution for looking at nutrients. I haven't mentioned turnaround time. Leaf testing we typically do in about 3 working days. And even our soil tests now, we have down to about 3 to 5 working days. So we're trying to get the results quickly back to you so you can use that data and get on what you need quickly. But we can only do as quick as we can do.
I'm happy to have another go if there's nobody else.
There's not a lot of action there, Dale.
Obviously, we use water in foliar application. Like we'll put nutrients into a tank and fill it with water and spray it out. Should we be testing our water?
Absolutely. I see so many issues here where people don't look at their water. Water test for agronomy and that sort of thing might only be $60 to $120 sort of for a full range of elements. You need to understand your water. I've seen serious water issues inland Australia, coastal Australia with salts. Bore water's changing as your aquifers change, as it gets drier or wetter. You can have breakthrough of different aquifers. So really understand your water, because if you do have salty water or high nutrient water or contaminated water, then it can be a serious issue for your sprays and definitely take away the effectiveness of a lot of what you're doing.
Thanks.
Very good. We've just got a question in the chat. Before I do that, if anyone's unsure how to raise your hand, it's in just at the top of the bar. There's a little hand that says raise, so you can press on that. So Maria's just posted in the examples you showed of the agronomy reports with the leaf tissue tests, were the conclusions on soil nutrient issues based just on the leaf tissue sample, or did they do a soil test as well?
In that particular case, they just based it on the leaf tissue for the comments in that report. However, a lot of these farmers that are doing these leaf tissues will often have a soil associated or at least some soil data as well for working out their fertilizer regime and what they want to put on. There are, like I said, there are agronomists, even your rural stores may be able to advise you on what nutrients you can add based on your leaf tissue test. And if it's showing deficiencies, they can also sort of work out some recipes or what you may need to add into your soil to fix up those problems with leaf. But typically a leaf tissue, a leaf sap or a leaf soil independently assess those reports and look at what nutrient issues you got there based on that particular result. But like I say, a lot of them have soil tests as well. Hope that answers it.
Thank you. Lee, you've got your hand up. And you're on mute.
Yeah.
You've gone to mute again, I think. Sorry, you're on mute. I can't hear you anyhow.
I'm back. Sorry about that. Thank you, Graham. Let's say we're running a multi-species for grazing, and we're just trying to get an overall picture of how it's tracking, what's cycling, etc. are we picking a single plant to sample, or are we sending an aggregate sample of everything that's in that mix?
Typically, I would say most of the time you're sending an aggregate. And a lot of people that are doing that are monitoring themselves from year to year or paddock to paddock,
Yeah.
and then really looking at the different guidelines. When you start looking at some of those pasture guidelines, you may look at 3 different species or 4 different species, and they may not be that different, particularly between different pastures. There's not a huge amount of data out there, so I think even in an agronomy report, they'd be able to work out what guideline to put to it. And you just say a mixed pasture. So the agronomist would probably have some fertility report and say, oh, I normally pick a particular species that matches well with a mixed pasture.
Yeah. And is there a reasonable database somewhere just to become aspirational on what pasture could be? If we're getting these tests back and you got all your numbers, can we see where we could head with that as for quality feed?
Yeah, for sure. And that's what a lot of our clients do. They look at what their best is, they get that data and then hold that up for themselves. But also a lot of the data you're getting from the fertility report is really trying to aim that ideal level. Not max is always ideal. So you want to be careful. Obviously, good protein levels, you want to hold that, but you want to hold the rest of your elements at the same time. I do find, and then when I've been down in New Zealand, they had some serious issues over many, many years where they were putting on so much urea. The pasture was a beautiful green, totally out of balance, and the grazing weren't getting the nutrition they require just because it was so unbalanced on urea. It looked fantastic. The whole system looked great, but it was a failure.
Yeah, well, that's kind of what I'm driving at. So in your guide, what you showed, you've got your guide. So is that what if we were getting close to those guides that you're showing us, is that close? Is that getting up?
That's the goal.
Right.
Yeah. So they have
Right.
excess levels, you have deficiency levels,
Yes.
and you don't want to be aiming in that excess level. You want to be aiming in those sufficiency levels, and they have quite a lot of levels there. So it gives you a good thing to work towards, and you'll really see where you've got toxicity issues and elevated. And like I said, once you're out of balance, I think it's not good for anyone. So
no, so that data that you've got there, what's that? Where's that been built from?
I'm not going to put up that reference, CSIRO.
So that's all come out of
all that's for dry tissue. For the SAP analysis, I will say quite clearly that I don't think there's anywhere near as enough SAP guidelines out there. The guidelines out there are basically on the web for SAP analysis and shared between a lot of agronomists, and they don't have a lot of basis to them. So if you just be really cautious with SAP analysis on interpretation and the guidelines, even from us, we give you numbers, but I can't stand by those guidelines. The soil analysis has the CSIRO data book behind it. For the leaf tissue analysis. And soil analysis has 100 years of data behind it, but SAP doesn't. It's a lot newer. I imagine some agronomists will claim they've got some really good data sets they've built up themselves. We don't have access to them. So just be a little bit careful with the SAP analysis.
Cheers.
Thanks.
We've just had a question typed in from Dale. Can you share any long-term trends you are seeing with soil analysis or leaf analysis over time?
I probably not on this presentation. It'd take me a while to sort of go through that. That is a real complex question. Obviously, with soil analysis, we want to see the carbon growing. And it's very hard to grow carbon, but we have a lot of regenerative farmers and good farmers, not necessarily regenerative, just farmers that are really working their soils well, getting good ground cover and that are building carbon. When you're building your carbon, you're building often your cation exchange capacity, which is building your nutrient level. And with leaf analysis, once again, as per that previous question, we don't want to be continually building nutrients. I've seen some studies by CSIRO that looked at leaf tissue testing and said, oh, we've done a fantastic job in farming over the last 50 years because we've increased the nutrient content of our leaves. But I think it's because we're doing so much feeding of salt nutrients or excessive nutrients in some cases that we're building up the leaf higher than what it should be. So I think we've got to be careful with showing trends over time. What I really look for in soil and leaf analysis is constant levels, not high levels and not dropping levels. So when you're looking at trend analysis in soils and leaves, you don't want to see it going along nicely and then plummeting or continually heading down. So you want to look at your trending so that it maintains or increases to some extent, but really your trending is to looking for where it could crash. And when it crashes, it's a long way to build it back up again. And you've got a lot of crop issues. And obviously, if your plants aren't performing or healthy, then it encompasses disease coming along and wiping you out, that sort of thing. So I think it's about balance and just looking at those trends to make sure it doesn't crash. So hopefully that helps, Maria.
A question from Brandon. Are soil biology tests available to show fungal and bacterial content?
Yes, we have a number of soil biology tests there that we offer. Still not to where I want. We have some with plate counts. We have some with PLFA, which is a fatty acid analysis. We work with a number of different labs and in-house on different biology tests. I'm looking at starting a PhD soon in my retirement years to move into studying soil biology and around Australia. And very keen on some enzyme tests for looking at soil biology. And there's some other DNA type tests that are coming forefront as well. But we haven't got all the answers there. There's some we can offer. However, the biggest note I'll note there, well, biggest comment I could say there, is when they've looked at a lot of soil biology tests, the biggest correlation they find is with the organic carbon. So your organic carbon is your feeder for your biology, it's your home for your biology, and it's where the biology really activates. And as a total biology, whether it's bacterial or fungi, it needs that carbon there as a feed source. So your carbon nitrogen ratio, I think are your good sort of basic biology tests that you get in a routine soil test.
Very good, thank you. I've got a question just on the washing of leaves. Is that something that we can do before sending it to yourself? Or if we think they're dirty, we should be asking the lab to do a clean?
I'd probably be cautious there on having people do it because you'd really want to know your water quality. We use ultra pure water that's been triple distilled and whatever else. It's called millicue water to make sure we can't contaminate those leaves, and we make sure we don't over soak them because we don't leach nutrients out of the leaf. Then we use a detergent to pull off any dust and that sort of thing. And it's a very special detergent that we use that is very, very low concentration, so it can't contaminate the leaf. So I'd probably be hesitant to wash yourself. And yeah, just be aware of it.
Yep, thank you. And you said in the presentation that most people are looking at doing a test in autumn and in spring. So if we were looking at using these tests to govern the nutrition that we were putting onto crops and pastures, would we just use that singular test? It tells me I'm deficient in this. My agronomist or myself think that we are going to put on these nutrients, and we just do that the whole way through? Or should we be retesting at some point within that time frame?
Look, we do have clients who will test autumn and spring to look at their uptake of nutrients. And when I do say autumn and spring, we get samples all year round. We don't have a quiet period in the lab, so there are people sending samples, whether they're leaf, sap or soil, middle of winter, the middle of summer. So Australia's a big country and it's got a very variable climate. So even though I was generalising there, a lot of clients are doing all different things, and I can't really speak for all of them. It's really what works for them and what they find is beneficial. And really, like I said, farmers are turning into researchers and scientists. They're doing their own research and saying, well, I've put on all this. What's the effect? what's my immediate effect? What's my effect in 2 months' time? So if they want to spend $60, it's not a big cost, when often they could be talking thousands or hundreds of thousands of dollars of fertilizer applications in the big farms, to try to work out what's going on and try to understand themselves.
Yeah, very good. Is there much of a difference in terms of what that plant's doing with nutrition? Say we've just sown a pasture or a crop, we're at the early vegetative stage, and then it goes through into later vegetation or heading. Do we need to do some different tests as it changes in its reproduction and growth?
It's the same test, so it's the same PA pack that we do, but you will see it vary quite a lot. The nutrient uptake, those mobile elements, can change very, very significantly in the different part of the plant or the different growth stage of the plant. And that's why a lot of the guidelines, when you send in a plant, you've got to note, oh, this is an early stage, or at what stage the plant is where those leaves come from. And that's so the agronomist with the fertility report can give you the proper guidelines depending on what part of the plant it is and what growth stage that plant is up to. Obviously, it doesn't normally matter that much in a pasture. It's mainly related to grapes and a lot of your cropping. Or your tree crops, that sort of thing. But yeah, you've got to understand what part and what growth stage you're in.
Yeah. I've just got a question from Maria. Can we use our tissue testing to indicate whether nitrates might be a problem for animal feed, or does that just need to be done on a feed test basis?
No, it's the same test. The nitrate is the same test. So when we do a hay or something for nitrate, we're just looking at that same nitrate test. So yes, you can use that. It's very interchangeable. So, and it gives you that good data. And obviously the cyanide as well. If you needed to do that separate test, it's another test that's available.
Yeah. She's also asked, do you use different agronomists for reports that have different plant specialties, or is that up to an individual to decide on?
Pretty well up to an individual. Like we said, this is a third party we offer. We may build up more third party reports as we look into it more and see what's out there. But you can take any sap, leaf or soil result to any agronomist and they can interpret for you. Typically, most of them around Australia know our results and know our format, so that they can do the interpretation for you based on these results. So you don't need to restrict yourself, and once you've got those numbers, you can share them around to different agronomists and get different opinions. That's quite interesting sometimes, how many different opinions you can get on the same set of numbers. But I spoke about errors before. A lot of farmers obviously find their agronomists they like, but often what we try to teach people and the farmers is look at the numbers yourself. Don't just rely on the agronomist. Look at the soil. We give soil guidelines for different soil types. And you have these guidelines, or I can photocopy pages or scan pages out of the plant CSIRO guidelines and send to clients as well. We've got no problem sending that sort of information through on a personal basis. And yeah, learn yourself. Don't just rely on others. And I think farmers have generally learnt that lesson already.
Very good. Well, we might leave it there. Thank you very much, Graham. I'll just share my screen again so you can all have another look at those dates. As I said, we'll get those emails out to you and hopefully you can come along. But thank you all for coming this evening and listening to Graham. Thank you so much, Graham, for your wealth of information. And I suspect you might be getting a few emails come through with questions.
I'll try to keep up with them.
Yeah. So, and if anyone does have questions from this webinar, they can send it through to us, and we can send them in a bulk load and bring them back to everyone. Otherwise, we will have this recording posted on YouTube as soon as we can for people to watch. And thank you all. And please do the feedback form.
Thank you, Hannah, and thank you, everyone. Catch up next time.
Excellent. Excellent. Thanks, guys. Have a good night.
Thank you.
Cheers, Graham. Cheers, Hannah.
All right. Bye.
Thank you.